Assalamualaikum and hello everyone. We are students from UiTM Tapa, and in this video we will be presenting about topic from CHM256 subject, and the topic that we chose is paper chromatography, but our main topic is thin layer chromatography, separation of nucleotides and nucleosides on silica gel. Alright, before we move to the next part, let me introduce you guys to our group member. The first one is me, Amalina Nasuha binti Azman, and Haris bin Azmin, Majda Nabil, and Ain Farisa. And this is the table of content on what we are going to present in this video. Alright, so in this experiment, we will explore how thin layer chromatography is used to separate nucleotides and nucleosides. But before we dive into the experiment, let me tell you guys what is TLC. TLC is a separation technique in which a sample is applied to a stationary phase, and in this case, we use silica gel as the stationary phase. And the mobile phase, a mixture of isopropanol, water, and ammonia, carries the sample up to the plate and separating its component based on their affinity for the stationary phase and their solubility in the solvent. Now, let's turn our focus back to the main experiment, which involved the separation of nucleotides and nucleosides on silica gel. This technique was first demonstrated by Sledman et al. In 1984 for separating nucleotide and nucleoside derivative. Nucleotides such as ATP and JTP are composed of a phosphate group, a sugar, and a nitrogenous base, whereas nucleosides like adenosine and guanosines consist of only the sugar and the nitrogenous base. This experiment highlights the power of TLC, a technique that separates a compound based on their chemical properties like polarity. By using a silica gel plate as the stationary phase, we can observe how different nucleotides and nucleosides move at different rates depending on their interaction with the silica gel and the solvent mixture. This allows us to analyze and identify this important compound in biochemical research.
Hello, my name is Majda. Now, before we step right into the procedure, let us go through with the materials and reagents needed to carry out this experiment. Starting with the nucleotide standards, which are the adenosine 5' monophosphate, uridine 5' monophosphate, cytidine 5' monophosphate, and guanosine 5' monophosphate. Next is the nucleoside standards, which are the adenosine, uridine, cytidine, and guanosine. Next, isopropanol water ammonia mixture. This is going to be our mobile phase. Silica gel plates, the stationary phase, glass TLC tank, micropipette, and a shortwave UV lamp. Now, let us begin with the procedure for the TLC separation of nucleoside and nucleotides. Step 1. Preparation of standards. The purpose for the preparation of standard is to prepare individual solutions of nucleotides and nucleosides at a concentration of 3 microgram per microliter. Firstly, use an analytical balance to measure 3 mg of each nucleotide and nucleoside. Then, add 1 ml of deionized water to the weighed compound in a clean tube or vial. Next, vortex or shake until the compound is completely dissolved. Lastly, clearly label each solution with the compound name and concentration. Store appropriately, approximately at 4 degrees Celsius. Step 2. Application of samples. Using a micropipette, apply 5 microliter of each solution onto a pre-adsorbent silica gel plate. Each sample is applied separately to avoid overlapping. Step 3. Development. Firstly, equilibrate the glass TLC tank with the mobile phase isopropanol water ammonia mixture with the ratio of 7:2:1 for 10 minutes. Then, insert silica gel plate and allow it to develop for a distance of 13.5 centimeters past the starting line. The development process could take up to 3.5 hours, during which the compounds will separate based on their polarity. Step 4. Detection. Once the development is complete, visualize the separated compounds under a UV lamp. The silica gel plate contains a fluorescent indicator that activates at 254 nanometer. The compounds will appear as distinct spots where the fluorescence is quenched. Step 5 and final, data collection. Measure the RF values of each compound, which helps in identifying them and studying their characteristics based on how far they travel relative to the solvent front.
Alright, now let's talk about the benefits of using TLC for this separation. The first benefit is simple and cost effective. TLC is an inexpensive and easy to use technique and it doesn't require costly equipment, making it ideal for educational labs or those with limited resources. The setup is straightforward and doesn't require highly specialized knowledge or training. The second benefit is efficient separation. Silica gel is an excellent stationary phase, providing effective separation of nucleotide and nucleosides. These compounds differ in their polarity, and TLC take advantage of these differences to separate them efficiently. Alright, and finally, the last benefit is time efficient. Another advantage of TLC is that we can analyze multiple samples on a single plate, and this makes it a time-saving method as we can quickly process several compounds simultaneously, saving both time and reagents.
Hello, my name is Haris bin Azmin, and I will be talking about the uses of separation of nucleotides and also nucleosides. First, it is used to monitor the chemical reaction. Next, we can use it for purification and also for identification. To elaborate, this method can be applied by applying the samples at different stages. This way, researchers can observe the formation of the product by observing the movement of the spots on the chromatogram. Next, TLC can be used to purify nucleosides and also nucleotides from complex mixtures. This is because TLC enables pure separation by exploiting the differences in polarity. Next, this method can be applied to identify specific nucleotides and also nucleosides by comparing their RF values. This way, more research can be made to study about genetic analysis and also metabolic pathways. Next, I will talk about the results and discussion. E represents adenosine, H represents guanosine, F represents uridine, G represents cytidine, A represents adenosine 5-monophosphate, B represents uridine 5-monophosphate, and C represents cysteine 5-monophosphate, and D represents guanosine 5-monophosphate. The RF values for each compound is E is equals to 0.63 and H equals to 0.53 and F 0.48, G 0.44, A 0.25, B 0.23, C 0.13 and D 0.09.
The highest RF value is 0.63, belonging to E, adenosine, indicating it traveled the furthest on the TLC plate. Conversely, the lowest RF value is 0.09, associated with D, guanosine 5-monophosphate, meaning it moved the shortest distance. RF values are crucial in thin layer chromatography as they help differentiate compounds based on their interactions with the stationary and mobile phases. By comparing these RF values, researchers can potentially identify unknown compounds in a mixture. However, it's important to note that RF values can be influenced by various factors, so they are not always absolute and can vary under different conditions. In conclusion, the experiment successfully separated and identified nucleotides and their corresponding nucleosides using the thin layer chromatography or TLC on a silica gel plate. Through this process, we observed that the nucleotides had lower RF values compared to the nucleosides. This suggests that nucleotides interact more strongly with the polar silica gel, likely due to their additional phosphate groups, which make them more polar. These observations demonstrates how polarity plays a significant role in chromatographic behavior. The TLC method proved effective in resolving mixtures of nucleotides and nucleosides, helping us distinguish between these two components. However, to improve the accuracy of future experiments, it's essential to take a few precautions. First, TLC plates should be handled gently to avoid cracking or breaking as they are fragile. Second, direct contact with the silica gel should be avoided to prevent contamination or altering its surface, which could interfere with the results. By following these precautions, we can ensure more reliable and accurate chromatographic results in future experiments.
Thank you for watching.